Now we are going to solve question from section B, which are from 2 marks questions. And the first question is question 17. Name a genetically modified cereal crop having enhanced nutritional value. This is for question 1. And the question 2, the nutrient it is rich in. Now we can give any cereal crop, that is a grain crop. So genetically modified cereal crop, we are giving the golden rice. This is example of genetically modified crop. And the nutrition value, it is rich in vitamin A. So we know that vitamin A, that is content which improves our eye vision. So this is the first question for A. Question B, state any 2 benefits of genetically modified crop. So genetically modified crop are very better than net pure variety. So we have benefits here. You can give only 2 answers. Crop benefits which are become more tolerant to abiotic stress. Abiotic stress like cold, drought, salt, heat, and any kind of soil quality. And it also helps to reduce post-harvest losses, which we can usually see in pure variety. Reduces reliance on chemical pesticides, or they are pest-resistant crops. So genetically modified crop are very better than pure variety. Now, by using Punnett square, depict the genotype and phenotype of test cross, where green pod color capital G is dominant over yellow pod color small g in garden pea with unknown genotype. Now we know that test cross is between the homozygous recessive and homozygous dominant, and after that we have to cross with homozygous recessive and heterozygous dominant. Now the test cross where an individual is showing dominant phenotype but unknown genotype is crossed with the homozygous recessive parent in order to identify the genotype of the individual is called test cross. So we have to show this through Punnett square. So the Punnett square is ready here. First we are going to do the cross with homozygous recessive and homozygous dominant. Now here the result will be all are going to be green. Now we have genotypic and phenotypic ratio, all are green. So here all are green, and we got now new parent, that is heterozygous dominant. And now in case 2, we are going to do cross with homozygous recessive, that is small g and g, and heterozygous dominant. Now we here got here also we can see 1:1 type of like genotypic 1:1 and phenotypic 1:1. That means here 1 green and 1 yellow or equal type. Like there is a 4 says, then we can see 2, 2 like this. We can find equal amount of homozygous recessive and heterozygous dominant plant. Here we have certain specific bacterial spores are mixed in water and spread over brassica crop to control butterfly caterpillars. So name this bacterium and its mode of action on the butterfly caterpillar. We have to give name of bacteria. So here the bacteria which used to control or reduce the butterfly caterpillar in brassica crop is Bacillus thuringiensis. Now it have the mode of actions. Now here we have the mode of actions here. Bacillus thuringiensis forms protein crystal during a particular phase of their growth, and this crystal contains toxin insecticidal protein, which is inactive protein. And when the butterfly caterpillar larva feeds on the plant, the spore containing inactive protoxin is reach the gut of the larva. Now another action, that is inactive protoxin is converted into an active form of toxins due to alkaline pH of the larval gut. This is how this works. Activated toxin bind to the surface of the mid-gut epithelial cells and creates pores. This causes cell swelling and lysis and eventually death of the larva. This is how these bacteria act on the brassica plant, and this act butterfly caterpillars, this reduce butterfly caterpillars in the brassica plant.